Good morning, student. Today we are going to study post-transcriptional control of gene expression, RNA interference. To understand that, we will discuss some of the experimental evidences and then we will come to what is the meaning of RNA interference. Now, if you see this diagram carefully, you will see inside a cell, a transcription is going on and followed by translation also going on inside the cytoplasm. Transcription is going on inside the nucleus, where after that, the mRNA is coming outside inside the cytoplasm, and then followed by translation is going on. For years, molecular biologists have been using antisense RNA to inhibit expression of selected genes in living cells. What is the meaning of that? If you see this diagram carefully, red color molecule is showing here mRNA, and if you can design an antisense RNA of that particular mRNA, so they can form a hydrogen bond, and because of that it can bind and it can inhibit its expression. That means that mRNA is not now available for protein synthesis. So this is one step where you can silence, you can inhibit the gene's expression. So scientists have tried by using antisense RNA. Then what they have done, they have found that the sense RNA into cells worked just as well as antisense RNA in blocking expression of a particular gene. To a surprise, they have also found that if instead of an antisense RNA, if we insert or if you introduce one sense RNA, that means the same thing, that also somehow suppress or inhibit the gene expression. The reason was not known that time. Later, when they introduced double-stranded RNA, what they found that the gene expression level reduced remarkably. So later identified that for these former two cases where they are using antisense RNA and the next where they are using sense RNA, both the solutions there was a contamination of double-stranded RNA. So ultimately what they found, that double-stranded RNA work better than both antisense as well as sense RNA. So this was the effect of double-strand RNA, which actually causing the decrease of this gene expression, or it inhibits the gene expression. Apart from that, we also know that RNA has different mechanism to regulate the gene expression. There are some example. 6S RNA of E. coli binds sigma 70 of RNA pol and regulates transcription. The concept of riboswitch, which control metabolic operon. If you remember that secondary structure of RNA actually inhibit, because of this secondary structure they mask the internal sequence. Because of that the transcription was regulated. That's called riboswitching. And one remarkable example we can take, the secondary structure of tryptophan gene, the tryptophan operon, which determines RNA polymerase continue or attenuate. So these are the mechanisms where people already knew that RNA can play a role in order to regulate gene expression. Let's come to another finding. One of the first example was an attempt to intensify the purple color of a petunia by supplying extra copies of the pigment producing gene. Try to understand what was the goal of this kind of experiment. We are going to regulate the gene expression by introducing RNA molecule inside the cell. They started with antisense, then the sense, then the double-strand RNA. They found that for double-stranded RNA, the intensity of the function, that means the gene silencing, is more effective compared to other. Now here what they did, they introduced more copy number of an RNA inside a petunia flower. You can see the flower here. What was the intention? To intensify its pigment. So more I express the gene, the basic concept is that the more gene will express the pigment gene, more colorful flower will blossom. But to their surprise, what they found, that instead of increasing the color, what they found, the 25% of this transformed plant, the flowers either they are white or they are coming with a purple color with white patches. So that was a surprise, a result for them, that how come when we are introducing a gene, how come that is suppressing on the other hand. That gives an idea that double-stranded RNA definitely playing a crucial role in gene silencing. So that's they named as the other name of the post-transcriptional gene silencing, PTGS. That's what these are all post-transcriptional. That means after transcription gene silencing. They named as gene silencing. It also referred as RNA silencing, as RNA is playing a critical role. RNA interference in some fungi, that is also named as quelling. RNA I is in a small letter, I stand for interference, can spread throughout certain organism. We will see what is the mechanism behind it. So far, what is the information I wanted to tell you, that RNA, double-stranded RNA plays a crucial role in gene silencing. That's post-transcriptional gene silencing. Let's come to the mechanism. What is the function? What are the steps involved here? In 1998, Fire and colleagues, as well as other scientists, also demonstrated that RNA interference is a post-transcriptional process that involves mRNA degradation. That means gene silencing, meaning which level silencing is happening? They cause the degradation of mRNA. So what is I'm giving? I'm inserting a double-stranded RNA. As a result, what is the output? The targeted gene, which gene I'm looking for to silence, that mRNA is degradation taking place. Now, the double-stranded RNA had to include exon region. Now, the double-stranded RNA, which from the beginning I'm telling scientists are introducing inside the cell. Now, how to choose which part of the double-stranded RNA we are going to choose? Which segment? Because from the DNA, you know that we are talking eukaryotes. So eukaryotic DNA gene sequence, if you think, there are intron, there are exon, there are promoter sequence, all these structures are there. So among this one, when the first transcription takes place, you all know everything comes. Then the splicing takes place. So when we are talking here, we will design a double-stranded RNA based on which sequence. Now, the double-stranded RNA had to include the exon region. Double-stranded RNA corresponding to introns and promoter sequence did not cause RNA interference. Then, what is the meaning of this sentence? That means the gene which I am going to silence, I have to design a double-stranded RNA of that gene from which part? From the exonic sequence, not from the intron, not from the promoter sequence. Now, the effect spread throughout the whole organism. After that, we will see in each tissue, each cell, that particular gene against which we have developed the double-stranded RNA, that mRNA will be degraded. Several investigators reported the presence of short piece of double-stranded RNA. They call them short interfering RNA or siRNA in cells undergoing RNAi. Now the next line is that when we have developed double-stranded RNA and that we are introducing inside the cell, hoping that my target RNA, the gene which I am going to silence, that mRNA will be degraded. In between the process, inside, scientists have observed when this process is going on, small RNA some small double-stranded RNA are forming. So from where they are coming, we will know that, but then referred that is siRNA. That is the meaning of short interfering RNA. So in between this mechanism, some small short interfering RNA is synthesized, that is named as siRNA. Now a small glimpse of the mechanism. There is a nuclease. There are not only one nucleus, two nucleases takes part here. A nuclease digests the trigger double-stranded RNA into fragments about 25 nucleotide long, and these fragments then associate with the nuclease, with this is second nuclease, and provide guide sequence that allow the nucleus to target the corresponding mRNA. Let me explain it. So what is there? I have a target mRNA, which gene I want to silence. Understood? Then for that, I have designed a double-stranded RNA. From which part I have designed the double-stranded RNA? From the exonic sequence of the particular gene. So that double-stranded RNA, when introduced, it comes, and this double-stranded RNA, one nuclease actually target this double-stranded RNA and make them small pieces. Okay, small double-stranded pieces. So this small double-stranded pieces we call short interfering RNA. Now this small double-stranded piece of RNA, which we are generating of 25 nucleotide only, after this nuclease they digest it, this small double-stranded RNA now go and bind my target mRNA. As it is a double strand, one strand is definitely complementary to the mRNA because I have designed such a way. So it will go and bind, and with the help of another nuclease, it will degrade the mRNA. So this is the glimpse of the mechanism. Let us see. RNA interference can be induced by many ways. I started by telling introduced inside the cell. But if it is depending on which system we are working, if it is a C. elegans, you can, this is the double stranded, the green and blue color, that part we are looking for. So we can feed the organism, you can grow that in the culture medium, we can inject it. If it is a Drosophila, inject embryo with double strand RNA. Most of the cases, if you handle the cell line, we need to inject it, incubate with this double strand RNA. So these are this is the maybe the create the transgenic flies that express double-stranded RNA, which is in hairpin. Then you can digest it also. Now what? Cultured mammalian cells, embryonic culture cells or oocyte or early embryos, you can transfect or inject double-stranded RNA. What I'm discussing here, I'm discussing what is the way you can introduce the double-stranded RNA. Again, I repeat, the double-stranded RNA we're going to design corresponding to the gene, from which gene I want to silence. Okay, so that exonic part of that gene, we need to design the double-stranded RNA. Now this is a hairpin loop. Different culture cells of whole organism transfer or synthetic SI. You can design also small RNA, double-stranded RNA, that can be designed different way. Hairpin loop, you can digest it. Transfected plasmid expressing miRNA will come later. Precursor like stem loop structure to get the siRNA cell. So we can incubate the cell lines with the double-stranded RNA, or we can directly inject inside the cell where we want to see the effect. Now come little bit in details. What is RNA interference or post-transcriptional gene silencing? Now I started telling double-stranded RNA need to be directed against an exon, not an intron, in order to be effective. Again and again, same thing I'm talking so that you understand it. What is RNA interference? We are going to silence the one particular gene expression. That means our target is the mRNA to be degraded. How? We are introducing a double-stranded RNA specific for that gene exonic sequence. Homology of the double-strand RNA and the target gene mRNA is required. That's the reason we have chosen the exonic sequence. Now targeted mRNA is lost. After effect of RNAi, what I will see, the mRNA is degraded. The effect is non-stoichiometric, that is, small amount of double-strand RNA can wipe out an excess of mRNA, pointing to the enzymatic mechanism. I will show the enzymatic mechanism. That means even if you give a small amount of double-stranded RNA to degrade your target, it will be wiped out the entire mRNA. So it will amplify in between certain step automatically. We will show the mechanism also. Now, single-stranded RNA does not work as well as double-stranded RNA. We'll see why and how. Now come to in further details. In 2001, it is not very old mechanism. It's a recent discovery. We should not say recent, but 2001, like that only, early time or 2000s. This is the discovery. Hammond and colleagues reported that they have purified from Drosophila the enzyme that cleaves the trigger double-stranded RNA into short pieces. So I started telling that we need a double-stranded RNA which will go inside, and some enzyme nuclease I mentioned will degrade them in small double-stranded RNA, which we'll call siRNA. The big one, I'm not talking siRNA. After nuclease digestion, what I'm getting small double-stranded RNA, that I'm referring short, that is siRNA. Okay, so the enzyme they have purified from Drosophila, and which actually cleave the trigger double-stranded RNA into short pieces. They named it dicer. Need to remember the enzyme they named as it dicer because it dices double-stranded RNA up into uniform size piece. I told you also 23-25 nuclear small nucleotide containing small species, double stranded it will generate. Dicer is a member of RNase 3 family. So again, so the enzyme which they purified from Drosophila, which actually target or cleave the double-stranded RNA which has been introduced or injected inside the cell into small 25 to 23 nucleotide length double-stranded RNA. This enzyme named as dicer, DICER. It is a member of RNase 3 family. Dicer cleaves 2 nucleotide 3' overhang. That means we have a 3' side 2 nucleotide extra at the end of the double-stranded siRNA. That means after dicer treatment, I am getting short interference RNA, that is siRNA. Understood? And phosphorylated 5' end. Now, dicer also has an RNA helicase activity. Why it is required, you will understand later. So, it can separate the two strands of the Si it creates. That means now I have, after dicer treatment, I have small RNA which we named siRNA, and this enzyme has another activity, that is helicase, RNA helicase activity. So, the strand can be, you can unwind. Okay, now I try to understand this double-stranded RNA small was generated from the gene exon sequence. Definitely one strand is complementary to the mRNA which is synthesized from that particular gene. Understood? Now, dicer does not carry out the RNAi effect. What is the RNAi interference? That means final degradation of the mRNA is not being done by dicer. That thing is done by some other enzyme. I mentioned earlier 2 nucleases. The first one is dicer. The second one, once we get the Si, that means small RNA, that appears to be the job of another enzyme called slicer. Which resides in a complex called RNA-induced slicing complex, RISC, RICE. So dicer finished its work, it produced Si. Now once the Si are ready, Si can be separated, two strands can be separated because it has a helicase activity. Now I need another enzyme who will take the guide strand, that means which is complementary to the target mRNA, and will go and bind and start degradation of the mRNA. That's the function, that's the goal of this entire process. That enzyme we call slicer. Okay, and it's a complex of RNA induced silencing complex. It's a part of this complex. Now see this mechanism. If what I discussed so far, this is a double-stranded RNA. This double-stranded RNA you can synthetically develop. Sometimes some transposons also can create this kind of effect. Sometimes some viral infection also can cause this kind of effect. Now, what we want, we need the double-stranded RNA inside the cell. Once the double-stranded RNA inside the cell, who will come? Dicer, that RNA's 3 family. That enzyme will come, the yellow color one, and it will cut it around 23 to 25 base pair small fragment. If you see this photo, this is not 25 because of simplicity. They cut it in small fragments. Okay. Remember, it is 25 nucleotide small RNA that we call what? siRNA. And you already there is a 3' overhang of 2 bases. Now this is called siRNA. Once this yellow color dicer produce the small RNAs, that is siRNA, its job is done. Now the second function will be done by, I just mentioned, slicer. That is a part of rice. You see, it is a 2-strand RNA, small RNA. Out of that, red one is complementary to your mRNA. So my target is the mRNA against whom I have generated this double-strand RNA. Now that, as it has an RNA helicase activity, so it's separated. This small red color RNA strand which is generated from the previous one, we call the guide RNA. Okay, it will come and bind to your target mRNA with the help of this orange color one is the rice, and it will start degradation of the mRNA, and that's the way you can see the complete abolition of the gene expression. That means that mRNA will be completely destroyed and you will not get the effect, complete gene silencing, by using this double-stranded RNA. So for today, this much is enough. I will again start this explanation in my next class. Thank you very much.